This method can help answer key questions about telomeres, such as is telomeric non-coding RNA TERRA localizing and acting in cis at its telomere of origin or in trans at different chromosome ends. The main advantage of this technique is that it allows researchers to visualize TERRA molecules expressed from a single telomere in living cells. Demonstrating the procedure will be Claudio Pegorar and Nicole Bettin, two graduate students from my laboratory.
To begin, grow AGS cells according to the text protocol. Then, when the cells reach 50 to 60% confluence, transfect them with sgRNA Cas9 expressing vector and the MS2 cassette. The next day, replace the culturing medium with medium containing neomycin. After this, add 10 microliters of 0.25% trypsin to each well of a 96-well plate. Using a microscope and a marker, mark the position of each clone visible in the dish. Replace the neomycin culturing medium with enough PBS to form a thin film of liquid on the clones. Using a 10 microliter pipette containing 5 microliters of trypsin, slowly release the trypsin onto the colony. Allow the trypsin to detach the cells for 1 minute. Scrape the colony with the tip and suck it up into the tip. After this, transfer the cells from the colony into a well of the 96-well plate. Incubate the plate for 5 minutes at room temperature. Then fill the well with 150 microliters of